Hi class, this is your instructor, Skylar Huff, and we're now in the 24th chapter, Viruses and Subviral Agents. So as we begin this chapter, one thing I'll say to keep in mind is just make sure that you pay close attention to where we are in the chapter and with your textbook. Along that same note, keep in mind that this chapter begins the first three major chapters for your final exam. In that, most of your multiple choice questions will come from these three chapters. And there will be a selection of conceptual multiple choice questions that will come from the prior chapters for study, including chapters 1 through, of course, 23. Along with that, there is homework for your final exam. And if you have any further questions, please see me after class, send me an email, or, of course, just stop by during office hours. So in the meantime, let us get to where we are today. And as I've also stated, just let me know when you have any questions. So to assist you with the final exam, we'll go over a few things to, of course, in this lecture. So during the late 1800s, botanists discovered the cause, meaning, and I'll put it as being they were looking for the tobacco mosaic disease, and that is what began to stunt the growth of tobacco plants and give those infected tobacco leaves a spotted mosaic appearance. So as they did find that they could transmit the disease to healthy plants by daubing the leaves with sap of diseased plants. So it was in 1892 that Dmitry Ivanovsky, a Russian botanist, showed that the sap was still infective after it had been passed through porcelain filters that were designed to filter out all known bacteria. A few years later, in 1898, Martinus Bergenic, which was a Dutch microbiologist, provided evidence that the agent that caused the tobacco mosaic disease, had many characteristics of a living organism. However, it appeared that the infective agent could only reproduce within a living cell. So Bergenic named the infective agent virus, which comes from the Latin word, which of course means poison. So the tobacco mosaic virus was the first virus to be recognized. So in the early 20th century, scientists discovered other infective agents, like those responsible for the tobacco mosaic disease, that could cause disease in animals or kill bacteria. These pathogens were so small that they could not be seen with a light microscope. They also passed through filters that removed bacteria. So curiously enough, they could not be grown in laboratory cultures unless living cells were present, class. So the development of the electron microscope in the 1930s made it possible to see viruses for the first time. Most viruses that infect animals, plants, and bacteria were identified during the second half of the 20th century. So viruses cause many plant diseases and are responsible for billions of dollars in crop losses each year. You may recognize some of the human viral diseases such as herpes, influenza, and even rabies. You have probably received vaccinations for many childhood viral diseases, including the chickenpox, which I had because, of course, at the time there was no vaccine. Mumps was one of which, rubella, and even polio. So emergent viruses are new viruses that appear suddenly or move into new areas, such as the novel COVID-19, that coronavirus. And they may cause epidemics such as, or even pandemics. So HIV, the human immunodeficiency virus, which causes AIDS, is an important example, just as COVID-19 or the novel coronavirus. So one that is also recent was, of course, the Ebola virus and even the Zika virus. And that's what you're seeing, class, meaning the Ebola virus is right there in photo. So as I continue on, class, viruses make up a large component of Earth's biomass, and their huge numbers make up for the small size and weight. So viruses influence many ecological processes. For example, they kill large amounts of marine biomass each day, contributing significantly to the recycling of carbon and nutrients. Viruses adapt to their environments through evolution. They also influence biodiversity of other organisms by transferring their own genes into the genetic material of their hosts. Viruses can also transfer eukaryotic genes from one organism to another, crossing species boundaries. So the discovery of giant viruses has raised new questions about the structure, genomes, origin, and the phylogenetic relationships of viruses. So in this chapter, we'll examine the characteristics and diversity of viruses, as well as a small subviral agent being the prion. So although biologists do not consider viruses to be living things, we include them in the diversity unit of biology because they have some characteristics of life and because they impact all living organisms. So to help you out with this class, I'll keep this lecture, I will say, as concise and precise as I am able to do. So as it states, a virus is a small infective agent consisting of genetic material and a protein coat called a capsid. So that genetic material class, of course, would be nothing more than either A, DNA, or B, RNA. So of course, keep in mind, class, that DNA is double-stranded and RNA is single-stranded. So viruses are dependent on a living host, a living cell. Hence, they're referred to as being an obligate intracellular parasite. Viruses are. So they depend on that living host cell to, of course, survive and replicate. So the reason, of course, that most biologists view viruses as non-living particles is because they are not composed of cells. And as you go back to the first chapter of study, we learned that, of course, the cell is that basic unit of life. So if it is, in fact, that basic unit of life and viruses have no cells, of course, they cannot be alive. And the same class is the case that they cannot carry on metabolic activities or reproduce on their own. So with that, viruses range in size class from 20 to 300 nanometers. And of course, there have been discoveries class of huge viruses. So viruses infect all types of organisms, including animals, as we likely all know, fungi, plants, protists, archaea, and bacteria, and some even infect other viruses. So to say it one other way, class, the outside of a host cell, of course, a virus consists of a nucleic acid core surrounded by a protein coat. So of course, I just mentioned that it will be either DNA or RNA. And then of course, the genome of the virus is typically pretty small, with about 5,000 to more than 100,000 bases or base pairs. So as far as the capsid is concerned, that it has those subunits, and they determine the shape, meaning the capsid or capsomeres, which are subunits, determine the shape of the virus. So they could be what is known as polyhedral or helical. And then, of course, some may even be an enveloped virus, as we see here. So as far as A is concerned, what you're seeing there is a rod-shaped virus. That is the tobacco mosaic virus. And there in B, what you're seeing, class, is the adenovirus. And the adenovirus would be polyhedral. And with that, the capsid has a number of units, and it's a 20-sided polyhedron. And here, of course, in C, what you're seeing, class, is HIV, the virus that causes AIDS, the autoimmune deficiency syndrome. So yes, the virus is leaving a host cell, which you all see there in pink. The host cell is there in pink. And then with that, the virus is enclosed in that envelope, which is derived from the host's plasma membrane. So the viral proteins project from that envelope, and the capsid, of course, of the virus is shown there in yellow. That is the human immunodeficiency virus. And then in D class, we're seeing T4, the bacteriophage. You should remember this class from when we learned about DNA being discovered by way of course, Hershey and Chase. In the meantime, this is a polyhedral virus with, of course, a polyhedral head and that helical tail. And this, of course, is a virus that attaches to the cell wall of the bacterial host by its tail fibers. So quickly, class, let's do what is called envelope viruses, and we're just about done. So as far as the envelope viruses are, HIV is an envelope virus, and it's surrounded by that outer envelope of the plasma membrane that's derived from the host cell. So while inside the host cell, the virus, of course, creates its proteins and inserts them into the host's plasma membrane. And then, of course, the HIV virus produces that envelope proteins that extend out from the envelope as spikes that interact with the host cell. And you may sometimes hear those being referred to as viral epitopes. And of course, those are used for cell recognition, of course, to allow that virus to recognize those cells. And in the case of HIV, it would be for those CD4 cells. So there it is. And you've seen those glycoprotein spikes that I mentioned, class, for recognition, class, of the host cell. So those glycoproteins are there, and of course, in order to recognize those others of which to, of course, bind to. So next up is viral classification. The viruses are not at all classified into one of those three domains of life, being, of course, archaea, bacteria, and eukarya. They are, of course, characterized based on their host range, and, of course, things such as size and shape, or even the presence or absence of an envelope, and the method of transmission. So, of course, they're classified, class, not by the Linnaean system, but, of course, the ICTV and the Baltimore classification system. So it's the International Committee on Taxonomy of Viruses. That's one of which, and the other of which, class, is the Baltimore classification system. So I won't get into either of those more specifically because I won't test you on those, but that is how viruses are classified. Let's get into viral replication. So it happens, class, in, I would say, one of two ways. And as I get here, let's just move on now, class, to the two types of viral reproductive cycles, being the lytic and lysogenic cycles. Let's begin with the lytic cycle. So the lytic cycle, class, in this case, the virus destroys or lyses the host cell. And typically, if the virus only uses the lytic cycle, it is particularly virulent, causing disease or death. So, of course, the virus forces the infected host cell to use its metabolic machinery to replicate viral particles. And then, of course, viruses that only have a lytic cycle are virulent, and I said cause disease or often death. So let's get, class, now to those 5 parts of the lytic cycle. Number 1, class, is going to be attachment or adsorption. So the virus attaches to specific receptors on the host cell, and this ensures the virus infects only its specific host. And the second portion here, being number 2, is penetration. So the virus penetrates the host plasma membrane and moves into the cytoplasm. Many viruses that infect animal cells enter the host cell intact, and some phages only inject their nucleic acid into the cytoplasm of the host cell, and the capsid remains on the outside. So right here, number 3, what's their, class, is replication and synthesis. So the viral genome, class, contains everything needed, class, meaning all the information necessary to produce new viruses. So once inside the host cell, the virus degrades the host cell nucleic acid, and then the virus uses the molecular machinery of the host cell to replicate its own nucleic acid and produce viral proteins. So many antiviral drugs interfere with replication of the viral nucleic acid. At 4, it is assembly. So newly synthesized viral components are assembled into new viruses. And number 5 is release. Assembled viruses are then released from the cell. And it's generally, class, speaking, that lytic enzymes produced by the phage in late replication process destroy the host plasma membrane. So the phage release typically occurs all at once and results in rapid cell lysis. In contrast, animal viruses are often released slowly or put off from the plasma membrane. On to the lysogenic cycle. So in the lysogenic cycle, however, what happens here is that the viral genome becomes integrated into the host DNA. So with this, class, I would say that it's the case in which we have these temperate phages that they do not typically destroy the hosts, and they alternate between lytic and lysogenic cycles. And of course, the integrated virus is called a prophage or provirus when it, of course, is in the bacterial DNA and then it replicates. So this class shows that very same with the lysogenic cycle. So let's get on to viral diseases here briefly. So with this class, just keep in mind that it was one stated class that this is a piece of bad news wrapped in protein. And this that I just gave you class was stated by the 1960 Nobel Prize winner in Physiology of Medicine as defining a virus as such. So viruses that are pathogens are responsible for some serious diseases, including Ebola, that hemorrhagic fever, rabies, influenza, hepatitis, AIDS, and the novel COVID-19 or coronavirus. But do keep in mind, class, that most viruses, however, do not cause disease. And in it that most viruses, including those that normally inhabit the human body, are harmless, and they are symbionts. And interestingly, viral genes from retroviruses comprise 8% of the human genome. And some of those genes code for proteins that are essential to human life. So let's get to a few examples here. So the viral infections class that do not usually kill plants, but just stunt the growth and cause changes in the shape of the foliage, or causes spots, streaks, or even motile patterns in leaves, flowers, or fruits. So most plant viruses have capsids and not envelopes, and the genome of most plant viruses consists of a single-stranded RNA. So, excuse me, being shown to you now, class, is a virus-streaked tulip. So it affects the pigment class there in those petals. And then, of course, what you're also seeing in the right-hand side here, class, is pepper leaves infected with the tobacco mosaic virus. So, of course, the leaf is characteristically mottled with light green areas. So in plants transmission, insects are important vectors of plant disease, and viruses cannot penetrate the cell walls unless the cells are damaged. So the viruses can also be transmitted from generation to generation by the infected seeds or even asexual propagation of the plant. So once that plant has been infected, the virus spreads through the plant body through those channels called plasmodesmata. So viruses cause serious diseases in animals. So animal viruses, they cause things such as hog cholera, foot and mouth disease, distemper class in dogs, called canine distemper, and even certain types of cancer class. And human viruses cause chickenpox, which you all may not have ever had thanks to vaccinations, herpes simplex viruses, the mumps, rubella, rubeola, rabies, yes, warts, infectious mononucleosis, viral hepatitis, certain cancers, and even AIDS. So one thing that did stand out that I must also state, class, is that each year most people suffer from 2 to 6 viral infections. So just keep in mind that, yes, it's not all viruses, class. I repeat, it is not all viruses that, of course, particularly cause disease. Continuing on. So human activity, class, meaning these things that are contributing factors, such as, I'll say, global travel is a major contributor, and even urbanization. So it's not that people live so spread out, it's now that we are living closer together than people lived in the past. And even war contributes, of course, to epidemics of infectious disease. And then along that same note, class, living conditions also affect such as the level of healthcare, meaning especially in rural areas, the level of healthcare, class, is nowhere, I would say, near what's found, class, in many of those urbanized areas. And even, class, sexual practices. Then there again, physical stressors, including nutrition and even sanitation. So these things, class, contribute greatly to, of course, infections by way of some virus. And even, class, bioterrorism, meaning it's of critical concern. So since September 11th, 2001, with the terrorist attacks in the United States, bioterrorism has become a critical concern worldwide. So with bioterrorism class, that's that intentional use of microorganisms or toxins derived from living organisms to cause death or disease. So this could very well be how an epidemic class of smallpox or anthrax or even the plague, yellow fever, Ebola, the hemorrhagic fever, or other potential fatal diseases class can, of course, be begun. So rapid identification and effective treatments and development of deployment vaccines for these diseases continue to be of critical global mission for scientists and healthcare officials and even governmental strategists. So let's now get to emerging diseases. So an emerging disease, by definition, class is just what appears suddenly and are new to the human population, such as AIDS, SARS, and even Ebola. And it says this sector. And then re-emergent diseases are those that have been almost eradicated, then of course strike unpredictably, causing an epidemic. And sometimes this is even in a new geographic area, such as the polio disease. So H1N1 class is an example of such. As it evolves and mutates, its RNA reassorts in birds, swine, and humans. As this outbreak occurred class back in, I would say, back in the fall, I guess it would be in 2009, going into 2010. And this continues on class with, of course, COVID-19, the coronavirus, with its RNA genome. So this is class what it looks like, or the coronavirus, with its viral epitopes there on the outside. They're to attach to very specific cells, but it's in the coronavirus family. And I won't go through all of the taxonomy of this. What I'll further get to, class, is that it has a single-stranded RNA genome with over 29,000 nucleotides, which encodes class for an approximate 9,800 amino acids. Then, of course, with this class, 80% of the nucleotide identity is with human SARS. So this new virus is, of course, the SARS-CoV-2. So I would not say that this is totally new. It's just, of course, by way of those mutations and its evolution. So genetic analyses suggest that the SARS-CoV-2, this coronavirus class, is probably evolved from a strain found in bats, and that's probably. So I included this class for informational purposes, but also to provide any assistance or help. So this class was published in 2020, and of course this is from the Lancet. So what's here, class, is just information that reflects findings presented in a research article that was published February 15th, 2020, and reflects a small number of early cases from Wuhan, China. And of course, more recent information with updated figures may be available from the Lancet/coronavirus for the latest research. So this gives you a timeline of the virus onset. And this class has an asterisk. Of course, this is in days, but that asterisk class is the median time for onset of symptoms, including fever in approximately 98% of patients, a cough class in 75%, fatigue class in 44% in others. So they say that an approximate 7 days class that the person may or may not be hospitalized, and just over a week class after onset of symptoms is a shortness of breath, and then of course comes the acute respiratory distress syndrome. And depending upon the person class, he or she class may be admitted into an intensive care unit. So secondly, class, getting into what I showed there right before class, is the way in which the H1N1 evolves as it mutates and its RNA reassorts in, of course, humans, swine, and birds, and eventually becomes virulent. So what happens, class, is the influenza virus, that's, of course, not infectious to humans. Well, what then happens is it becomes a mutant version, meaning it changes. And as it becomes that mutant version, it then affects, class, can infect that human cells, but not spread to other humans. So this is what happens. And as it does happen, class, this is why it's just so important. So as that is the case, just keep in mind that we should remain vigilant and, of course, wash our hands and wash our hands well. And in the event that there is a mandate stating that masks should be worn, let's wear those masks if that is the case. So now get to viral infection in humans. So here, most viruses cannot survive long outside of living hosts. They must be transmitted class from one animal to another. And the host range may very well be limited. And just keep in mind that the poliovirus, which I previously mentioned, class, attached to the cell of the human digestive tract, and then, of course, the motor neurons of the brain and spinal cord. So it's just very specific. As I mentioned, class with HIV, meaning, of course, those CD4 cells. It has to have those CD4 cells. And it's very specific, as I said, host range may be quite limited because, of course, the attachment proteins on the surface of the virus are very specific, meaning those viral epitopes. And this shows something that is quite similar to class. And this is showing you all what happens class with an enveloped virus. So as the enveloped virus class has the receptors class on that target cell or that host cell, or the very same class, and those viral epitopes fit those like a lock and key. So then the membrane fusion class allows the viral envelope to fuse with the plasma membrane, and the virus class has now gained entry into the cell. So with that, the virus can then is released into the host cell's cytoplasm, and then the viral nucleic acid separates from its capsid, as you're seeing here, number 4. Here at 5, then of course, the viral nucleic acid enters the host cell nucleus and then begins to replicate. So as replication then occurs, the virus at number 6 class can then transcribe its nucleic acid into mRNA. At 7 class, it can then use those ribosomes. It can then use those ribosomes here at number 7 to, of course, direct mRNA to synthesize the viral proteins. And ultimate class at 8, I mean, this is a textbook definition class of the central dogma of DNA, from DNA to RNA to protein. But number 8 class, the vesicles can then transport the glycoproteins to host cell's plasma membrane. And then at 9, new viruses are assembled class and enveloped by host cell plasma membrane. And 10, viruses are released from the host cell. And this is what happens class in an unvaccinated child that has a rash, which is, of course, a major symptom class of German measles called rubella, which is caused by an RNA virus spread by close contact. So this class is just an example, but this is indeed, class, how it happens. So having done this class, I'll continue on. So with retroviruses, these are those RNA viruses that have DNA polymerases and, of course, reverse transcriptase. So what happens is reverse transcriptase transcribes the viral RNA genome into DNA, which is then integrated class into the host DNA by a viral enzyme. Then, of course, the viral genes are transcribed by the host RNA polymerases to, of course, make more copies of viral RNA. And then the viral structural proteins are synthesized, the capsid is produced, and new viral particles are then assembled. So viruses class like this that do not have an outer envelope, they exit the cell by lysis, and then the envelope viruses are picked up by a fragment host plasma membrane that is a lead infected cell. For example, class HIV is what they call retrovirus. So it uses reverse transcriptase class to, of course, create what is then the DNA and then go around the other way. So I cannot state enough, class. Those CD4 receptors are there. So as they are there, class, on the plasma membrane of the T helper cells, they can then, of course, get to DNA, and then from that DNA class, transcribe, of course, into viral RNA, and that viral RNA then leaves, of course, the cell. So, and I'll get to this stating again, it's there being one reason, class, that HIV is so virulent is because of the way it mutates. A single person, class, can have thousands upon thousands upon thousands, class, of differing strains of HIV. So, of course, that would mean that that person would need, of course, that many different drugs. Hence, class, many of those drugs are called ART, for ART therapy, so antiretroviral therapy, because antiretrovirals, of course, is to keep the viral load down, and of course, to keep the number of strains of the virus down to ensure that he or she, class, can, of course, live that life. And then, of course, resistant to antiviral drugs. So recent strains of influenza have become resistant class to amantadine, which of course is that drug that inhibits penetration or uncoating of the viral nucleic acids, and is probably due to use of in poultry feed. So influenza viruses are also becoming resistant to Tamiflu, which you all may have had before, if in fact class you've had symptoms of the flu, and of course caught those symptoms very early on. But what Tamiflu does is it inhibits class the neuraminidase, which is a viral enzyme necessary for the virus to leave the host cell. So of course, if the virus cannot leave, it of course cannot spread within the body. So at least now you know what Tamiflu does, be it class Tamiflu or the generic class for Tamiflu. So just keep in mind, class, that viruses, they do evolve, meaning as viruses evolves, excuse me, what happens is the viruses may have originated class as a mobile genetic element by way of plasmids or transposons that could be moved from one cell to another. And of course, they may have originated in animal cells and others class in plant cells or bacterial cells. Thereafter is the reduction hypothesis or the regression hypothesis that viruses evolve from a small independent cell. There are parasites in larger cells, and genes were gradually lost through evolution. So this is supported by certain bacteria that are able to reproduce only inside host cells, but explains why viruses are dependent on hosts. And with the virus first hypothesis class, it could be that they co-evolved with current cellular hosts even before life forms were assigned to the 3 domains diverged. In any case, class, just keep in mind that viruses do exist. So, and to end the chapter class with subviral agents, we'll be here, class, particularly with prions or prions. And as we are here, class, they were stated as, or called, class, the prion, as being those, I guess I'll say, infective agents that are smaller and simpler than viruses. So we won't do much with satellites or viroids. We'll get class to prions. So prions were discovered, class, by Stanley Prusiner. And they were discovered because the infective agent caused Creutzfeldt-Jakob disease, which of course is a degenerative brain disease that does not contain DNA or RNA. It's just that protein, class. So it's that proteinaceous infected particle. And this, of course, is also found in white-tailed deer populations, class, throughout this continent. In the meantime, the harmless protein PRP can sometimes fold into a new, different shape, the prion, and cause disease. And it's, of course, prions that aggregate and accumulate in the brain and other tissues that cause those fatal diseases. So they call this transmissible spongiform encephalopathy, and it's just the degenerative brain diseases of a mammal or even bird. So holes develop, class, just while they're called encephalopathy, and of course it becomes somewhat like a sponge, hence they're referred to as being those transmissible spongiform encephalopathies. So this includes, class, scrapie found in sheep and goats, and chronic wasting disease in deer and elk, and even the bovine spongiform encephalopathy, mad cow disease, which you all may have heard of, and of course I already mentioned, class, Creutzfeldt-Jakob disease. So this, class, has been the 26th chapter. Next up, class, is chapter 25, Bacteria and Archaea. So as we begin Bacteria and Archaea, class, just keep in mind that this is wholly different from the last chapter, which was on viruses. So here we will examine the diversity and characteristics of the members of domain Bacteria and domain Archaea, 2 of 3 main branches of the tree of life. These organisms, informally called prokaryotes, have been on the planet we inhabit, class, excuse me, class, for more than 3.5 billion years. Much longer than the eukaryotes, which evolved 2.2 billion years ago. Excuse me, class. So although prokaryotes are microscopic, they are so numerous that they probably account for more than half the Earth's biomass, meaning the mass of living material. Thus, the biomass of prokaryotes is greater than all of the eukaryotes, and that does indeed, class, include fungi, plants, and animals. So as scientists discover new prokaryotes and explore the new niches and habitats, the percentage of bacteria and archaea in Earth's total biomass continues to increase. So what had happened was, yes, I said that, was by Leeuwenhoek, that Dutch microscopist who discovered bacteria and other microorganisms in 1674 when he looked at a drop of lake water through a glass lens. It was then classed in the late 1800s that many microorganisms, including some bacteria, fungi, and protozoa, were identified as pathogens, agents that cause disease. Bacteria cause many diseases such as tuberculosis, tetanus, respiratory infections, and food poisoning in humans. However, class is only a small minority of bacterial species are pathogens. And although some archaea have been associated with the disease, no pathogenic archaea have been identified, class. So, as I say that, just keep in mind that these, being the bacteria class, are important for life on Earth. It is not just that they're here causing disease. So both bacteria and archaea play essential roles in the biosphere. As decomposers, they break down organic molecules into their components. Along with fungi, prokaryotes are nature's chief recycler. So without these microorganisms, elements such as carbon, nitrogen, phosphorus, and sulfur would remain locked up in wastes, and dead bodies of plants, animals, and other organisms would be unavailable for the synthesis of new cells and organisms. So one group of prokaryotes, the cyanobacteria, are producers that carry on photosynthesis, which generates oxygen. In class, that's what you are seeing in the picture, class, before you right now. So with that, it's that 70 to 80% of the oxygen, class, on planet Earth is generated by cyanobacteria and algae. Other prokaryotes convert atmospheric nitrogen to ammonia and then to nitrates, forms that are used by plants. Majorly important, class. This conversion enables plants and animals, because they eat plants, to manufacture nitrogen-containing compounds such as proteins and nucleic acids. So here in this chapter, we will describe the structure of bacteria and archaea, and then discuss prokaryote reproduction and evolution. But we also get a bit into the nutritional and metabolic adaptations of these organisms and describe the phylogeny of two prokaryote domains. We will then, class, present an overview of prokaryotic relationships with other organisms and the effect of prokaryotes on ecology, technology, and commerce. And then we'll discuss bacteria and disease to end the chapter. We're now here in the prokaryotes class. All right. So in contrast to viruses, prokaryotes are unicellular organisms, members of domains, archaea, and bacteria. So they're very small cellular organisms, about a tenth of the size of a eukaryotic cell. Most are unicellular, but some class form colonies or filaments containing specialized cells. And of course, I already mentioned, class, domain bacteria and domain archaea. The shapes in which class they come in, as you all have seen in lab, are cocci or coccus, meaning they are either singly occurring or in groups, maybe in twos, Diplococcus, in long chains, Streptococcus, or in clumps, Staphylococcus. Rod-shaped being bacilli or bacillus occurs as single rods or long chains of rods, and finally class spirals being the spirochete or spirillum. So they look, class, as a comma or spirillum as in Vibrio. So there they are, class: coccus A, bacillus B, and spirillum C. These are three basic bacterial shapes, class. So this class shows prokaryote structure. Note this figure, class, from the Fimbriae to the storage granules to the flagellum, the ribosomes, must have those for proteins. Bacterial chromosome, class, that's circular chromosome for DNA. The capsule, class, that cell wall, which of course has its outer membrane and that inner peptidoglycan layer, be it thick or thin. The plasma membrane, the nuclear area, it's no nucleus, class. It's referred to as the nucleoid, not a nucleus. And then, of course, the plasmid, that small ring of DNA class, allowing, of course, prokaryotes to change and gain antibiotic resistance. So the cell wall. The cell wall is what, of course, is around the plasma membrane, and it supports the bacteria, and it keeps the cell from bursting under hypotonic conditions. Yes, it includes peptidoglycan, that polymer that consists of two amino sugars linked to short polypeptides to form that single macromolecule that encapsulates and surrounds the entire plasma membrane. So you don't look for peptidoglycans, or at least that peptidoglycan, to be in an archaea. Archaea class, or at least members of Archaea, have no peptidoglycan layer. So let's get down class to classification of prokaryotes based on their cell wall. So let's get to gram-positive and, of course, gram-negative bacteria. So gram-positive bacteria have that thick peptidoglycan layer. So that thick peptidoglycan layer class is there, and the bacteria that is gram-positive absorb and retains that crystal violet stain, as opposed to, of course, gram-negative bacteria. So they have that thin peptidoglycan layer and that thick outer membrane. And here, they do not retain the stain. They do not retain that crystal violet stain. Gram-negative bacteria do not. So a number of prokaryote species produce a capsule, or that slime layer of polysaccharide or protein that surrounds the cell wall. So this is to protect the cell against phagocytosis. So without the capsule class, bacteria does not class cause pneumonia. So of course, they can also use the capsule class to attach surfaces such as the dental plaque on your teeth, plant roots, or even rocks. And with Fimbriae and Pili class, well, the Fimbriae and Pili are used to attach to cell surfaces or even to each other. So the Fimbriae are those hundreds of hair-like appendages that are shorter than flagella, of course, and they're made of protein. And the Pili are those appendages class, and there are longer, but then there are fewer than those Fimbriae. And the Pili class also help transmit DNA between bacteria. So the sex Pili. Yes, the sex Pili. On to now things known as endospores. So with endospores, these are extremely durable cells, and they are dormant. So what happens is they can survive in very dry, very hot, or even frozen environments at times when food is scarce. And as the conditions class become favorable, those endospores can then form that active cell. And of course, Archaea do not form any endospores. So if in that class the bacterium forms endospores, it is likely that that bacterium causes disease, such as in the form class of Clostridium tetani from tetanus, or Clostridium perfringens, or gas gangrene, or even class Bacillus anthracis or anthrax. So many types of bacteria are motile, and they can move by rotating their flagella class. That flagellum consists of three parts, which I want to ask you about, but it uses energy class in the form of ATP to allow them to move, much like a propeller. So Archaea also have Archaea also have flagella, which act as a propeller, but of course it's thinner class. And then, along with this class, just keep in mind that flagella do not move aimlessly, but they do exhibit class chemotaxis and can move towards food or towards one another. So flagella class that are not in prokaryotes allow them to move class by gliding or twitching. So there you are, class, the flagellum of bacteria. So let's get a bit into the evolution class of bacteria. So unlike the DNA in eukaryotes, or the eukaryotic chromosome class, here the prokaryote DNA has little protein. So in addition to which, it's genomic DNA. So the bacteria has plasmids. So you have those small circular plasmids of DNA that may contain genes that, of course, have catabolic enzymes for genetic exchange, or, of course, catabolic enzymes or genetic chains for antibiotic resistance. So plasmids replicate independently or become integrated in the DNA. So here with rapid reproduction, it happens so rapidly because they reproduce asexually, they being the prokaryotes. And it's done class by way of binary fission. One cell divides into two similar cells, in which, of course, the circular DNA replicates, and then you have that ingrowth of the plasma membrane, and then the cell wall forms a transverse wall. Budding also occurs, and in budding, the cell develops a bulge or bud that enlarges, matures, and eventually separates from the mother cell. Lastly, some prokaryotes divide by fragmentation. Here, the walls develop within the cell and then separates into several new cells. Nextly, gene transfer. So the gene transfer, this results in genetic recombination. In class, this is where it gets very interesting. So what happens here, class, is it can be vertical gene transfer, meaning transferring that genetic information from parent to offspring. Horizontal class, being genetic information class comes from another organism that is not its offspring. And this happens, class, gene transfer, in one of three ways. Let's hit those three now: transformation, transduction, and conjugation. So transformation, class, as it occurs, this is when you have bacteria DNA, plus, of course, DNA of another source. So the bacterial cell takes up DNA fragments of foreign DNA or RNA released by another bacterium, and then it exchanges that DNA in that resultant recombinant cell. With transduction, however, this happens when you have the bacterial DNA of one bacterium with the bacterial DNA of a second bacterium. In other words, class, a phage carries bacterial DNA from one bacterial cell into another, and then we have the chromosome of a new host that becomes recombination of its own DNA and DNA from another bacterium. Thirdly, class, is conjugation. So this is when you have plasmid DNA going through the bacterium. So you have two cells of different mating types that come together, and there is an exchange, class, of genetic material. So of course, this happens, class, and as it happens, as some call it bacterial sex, there you have it, the sex phyllis is there, all because of that plasmid. So with evolution of bacteria, class, the mutations, they occur and then they are quickly passed on to new generations, and effects of natural selection, class, are quickly evident. And it's all because of the way in which bacteria reproduce so rapidly. And then those changes, of course, can spread so quickly. So to kind of end things here, let's get to the nutritional and metabolic adaptations. So as it happens, the two main ways of obtaining nutrition, class, would be, of course, by being autotrophic, using inorganic compounds such as CO2 to manufacture organic compounds, or being heterotrophic, obtaining carbon atoms, class, from the organic compounds made by other organisms. So two main ways of capturing energy, class, being either chemotrophic or phototrophic. So chemotrophs obtain energy from chemical compounds, as opposed to phototrophs capturing energy from light. So being a photoautotroph, class, would include the cyanobacteria, and being a chemoautotroph would be, of course, using CO2, no sunlight involved. And being a photoheterotroph, of course, would be those purple bacteria that uses, of course, photosynthesis. So chemotrophs would include those decomposers or saprotrophs, and they harm hosts, causing disease. Some are beneficial, found in the large intestine, such as such as us and Homo sapiens, creating, class, vitamin K and vitamin B. Those B vitamins, class, and vitamin K, we must have those. So just keep in mind that those photoautotrophs include the cyanobacteria, taking the energy from sunlight, class, and taking in the carbon from those inorganic compounds. So we must have these. And of course, the chemoautotrophs, they take in the substances, class, such as the ammonia, and leave in the hydrogen sulfide. And then, of course, the photoheterotrophs, yet again, class, using the sun. And then, of course, I've mentioned also the chemoheterotrophs, class, taking in the decomposers, taking in the dead organic matter. And then, class, keep in mind that most prokaryotes do indeed require oxygen. So the aerobic bacteria require oxygen for respiration. Facultative anaerobes use oxygen, class, for cellular respiration if it's there. However, they can also use the anaerobic respiration when necessary. However, an obligate anaerobe, class, can only carry out anaerobic respiration. So of course, that final electron acceptor class is not going to be oxygen. And you learned that class back when we learned about, of course, the process of respiration. So nearing the end of things class, we're here with nitrogen-fixing bacteria, the rhizobia. So some bacteria and archaea reduce nitrogen in the atmosphere to ammonia, and then the bacteria convert that ammonia to ammonium ions used to produce organic compounds. And with nitrification, this is when I get to rhizobia, being that they convert ammonia or ammonium ions to nitrite, and then others convert the nitrite to nitrate, a form that can be used by plants and fungi. So we must have this nitrogen-fixing bacteria class. It allows the world to go around, I say. Plants must have it, and we must have plants to live. So really quick like class, the phylogeny of the two prokaryote domains, so archaea and bacteria, are fundamentally different. So we find them class here, seeing that first things first class was domain bacteria, meaning because of that common ancestor class to all living organisms. But here class it includes the proteobacteria with, of course, gram-positive bacteria, the cyanobacteria, the chlamydias, and the spirochetes. And then we get class to domain archaea, those extremis or the extremophiles. So archaea class have no peptidoglycan cell walls, and they have a simple RNA polymerase. So those two main phyla exist, but keep in mind that we find archaea class in harsh environment, such as the extreme thermophiles. That orange, yellow, and green colonies of extreme thermophiles thrive in Grand Prismatic Spring in Yellowstone National Park in Wyoming. And then the thermophilic red algae contributes to the green color shown here, class. So eukaryota includes the methanogens, the extreme halophiles, and the extreme thermophiles. So the methanogens class, of course, produce methane, billions of tons of methane each year. And of course, these are obligate anaerobes that produce the methane gas. The extreme halophiles will require large amounts of sodium for growth, such as the salt puns, the Dead Sea, and even the Great Salt Lake. So by aerobic respiration to make ATP, but also carrying out a form of photosynthesis using that purple pigment. And then, of course, we have the Karyota and the Nanoeukaryota. And the latter of which class were discovered in a hydrothermal event in 2002. They're small, they're anaerobic class, and extremothermophilic with very small genome. And they live attached to another Archaean. Then we have Archaean Supergroups class, that's TAC and then Asgard, which I won't get farther into. But just keep in mind, class, that the Archaeans are found in harsh environments, class. So we have major groups of bacteria class as mentioned. So there are mostly those that are gram-negative. The proteobacteria, the cyanobacteria, the chlamydias, the spirochetes, and then of course the gram-positive bacteria. So we have 5 major groups of bacteria. And then lastly, class, the impact on ecology, technology, and commerce. So it's a relationship class between these species. So mutualistic relationships class being that both partners benefits, and the one that's parasitic, being that one will benefit and the other partner class will likely be harmed. And I won't deal much class with commensalism at all. I will not deal much with commensalism. So let's continue on here. So with biofilms, so this is many bacteria in watery environments have those dense films, and many times class we find these in those microorganism communities class. And it's this that we also can find class with dental plaque, which consists of a film of bacteria embedded in a matrix of a glycoprotein, and that is what you also see in class in photo here. So bacteria in the plaque can produce acids that erode the tooth enamel, leading to tooth decay. And then ecologically class decomposes break down dead organic matter, which I've already mentioned. They're also class to fix nitrogen in the usable form. The nitrogen-fixing bacteria that transforms that atmospheric nitrogen class, and even that rhizobia bacteria class, part of that mutualistic relationship class between the legumes, and of course, the rhizobia bacteria. And then ultimately class the cyanobacteria, which takes huge amounts of CO2 into, of course, organic content used by class those others of which. And then commercially, we have the pharmaceutical companies that obtain antibiotics from the gram-positive soil bacteria. So it's not that we find an antibiotic class in a store necessarily. Antibiotic classes are found from the bacteria, typical class being that soil bacteria. And then, of course, genetically engineered bacteria produce vaccines. Also, things that are needed, such as insulin class and even HGH, called human growth hormone. And then bacteria also can ferment, making many foods and beverages. And then lastly, for bioremediation, in that they can remove oil or pollutants class from the environment. And then we can also use methanogens class in biogas production, but one big one class is using heat-resistant DNA polymerase derived from bacteria class found in those geothermal vents. So the Parochus class Furosis is using PCR for forensics and molecular biology because, of course, that DNA polymerase class can withstand those high temperatures. So to end things, class, we get to, of course, bacterial disease. So it's estimated that over 700 trillion bacteria class inhabit the human body. And even that normal microbial population class is what prevents harmful microorganisms from flourishing. So we harbor class many harmless symbiotic prokaryotes, microbiota and microflora. We must have these for life. So it's not that bacteria is wholly bad. I would say, class, if anything, bacteria is good. And we got to a point, class, in the 19th century where Pasteur disproved spontaneous generation by demonstrating that sterilization of a sugar and protein culture prevented bacterial growth. Also, class, led to that development of the rabies vaccine, showing that people can be stimulated to develop immunity to disease. And then, of course, in 1876, class, Koch demonstrated that bacteria causes infectious disease, which of course led to Koch's postulates that a pathogen must be present in every individual with the disease. We must have a sample of that microorganism taken from a diseased host that can be grown in pure culture, and then that sample of the pure culture causes the same disease when injected into the healthy host, and then ultimately, class, that microorganism can be recovered from the experimentally effective host. So even, class, from what was developed back in the 1800s is still used today, class. And then last, we get to adaptations to pathogens, or at least in pathogens. So to cause disease, a pathogen must be adapted to that specific cell type and be able to produce and multiply those toxic substances, such as Helicobacter pylori. That has an alkaline shield. Well, this is what causes those peptic ulcers. So it's that most common cause of those peptic ulcers in the stomach and duodenum, and it's an extremely successive pathogen. And it's also been associated with chronic gastritis, which of course is stomach inflammation with stomach cancer, the second most common type of cancer in the world, class. So as far as exotoxins are concerned, I'll be here, class, relatively briefly, but some bacteria produce strong poisons that are secreted or leaked out when the cell is destroyed. So with botulism, class, it's caused by an exotoxin that's released by gram-positive endospore forming Clostridium botulinum. So the botulinum toxin is inactivated by proper heating. And then with endotoxins, these, of course, unlike exotoxins, the endotoxins affect the entire body and are not destroyed by heating. And then we get to antibiotic resistance, which is, I say, huge. It's a major public health problem. So what happens is the plasmids, class, have genes for antibiotic resistance. And of course, the resistance to a specific drug or transferring the resistance to other bacteria. So some of our factors have several genes for drug resistance. Hence, class, if ever you are prescribed an antibiotic, you should take it all. I repeat, you should take it all. Or of course, it may be a point where things that were normally class cured by prescription of antibiotic begin to kill us because of the resistance, such as the case with MRSA and VRSA. So methicillin-resistant Staphylococcus aureus. And as I mentioned, such class, it's now class that it's resistant. So 1 in 100 healthy people, class, carry MRSA. And with that, MRSA causes more than 95,000 serious infections, resulting class in over 18,000 deaths. So this is quite an important class to know. And there's also a form called vancomycin-resistant Staphylococcus aureus. So it's all because of the overuse class of antibiotics. So I'm not saying you should not take an antibiotic. Just keep in mind, class, that those things, class, that are bacterial infections can many times, class, go away. And in the event that you take an antibiotic, just remember to follow the doctor's orders and complete that course of antibiotics. So that was the 26th chapter. Now on to class. Sorry, that was the 25th chapter. Now on to the 26th chapter, the final chapter, class, for study. Protists. So protists are an informal group of primarily aquatic eukaryotic organisms with diverse body forms, types of reproduction, modes of nutrition, and lifestyles. So protists, which include the algae, water modes, slime modes, and protozoa, are unicellular, colonial, or simple multicellular organisms that have a eukaryotic cell organization. So the word protist comes from the Greek for very first, reflecting the idea that protists were the very first eukaryotes to evolve. So with that being said, class, protists are members of domain Eukarya, the third domain on the tree of life, and they have eukaryotic cells that are characteristics of protists, as well as complex multicellular organisms to kingdoms fungi, animalia, and plantae. That's why they're sometimes taught as the animal-like protists, the plant-like protists, and the fungus-like protists. So with this, they, however, have a eukaryotic cell structure, and that is what clearly differentiates the protists from members of the prokaryotic domains, bacteria and archaea. And just keep in mind, class, unlike prokaryotic cells, the eukaryotic cells have nuclei and membrane-bound organelles, such as the mitochondrion, and plastids, or what is known as the chloroplasts, and even, class, the 9-2 flagella, and multiple chromosomes in which DNA and proteins form the complex called chromatin. So there is also what is known as sexual reproduction, class, by way of meiosis and mitosis being characteristics of eukaryotes. So until recently, class, all protists were classified in the kingdom Protista. However, class, that is no longer the case. So what is now done, class, is the classification system uses supergroups, which you all were introduced to, class. And with that, the classification, class, was by default, but of course protists include any eukaryotes that were not land plants, fungi, or animals, and based largely on molecular data that have clarified many evolutionary relationships among eukaryotes. Kingdom Protista is no longer a recognized clade, class. So although we have made great progress in understanding the evolutionary relationships among the protists, many of those relationships, class, are still unclear. So biologists currently recognize dozens of protist taxa, which, along with other eukaryotes, are classified into 5 supergroups. And of course, consideration of all protist taxa is beyond the scope of this text, class. So we'll discuss representative examples of those 5 supergroups and provide insight into such diverse prokaryotes may have evolved. So let's get there now. So eukaryotes. I've gotten to what makes a eukaryote. Let's get to diversity in the protists. So the body plan is quite diverse. Some are unicellular, some colonial. Some are coenocytic, and some are multicellular. So with that, most are multicellular protists with, of course, no specialized tissues. So most protists, class, were motile at some point, being able to move, and some of which, class, have pseudopodia, and others of which, class, move by gliding, waving cilia, or flagella, or even, class, flexing their cells. So the way in which nutrients are maintained, so of course, algae are autotrophic photosynthesizers. Some are heterotrophs, which obtain nutrients by absorption, other which ingest, and then some protists can switch modes of nutrition, being autotrophic, and then of course being heterotrophic. So many protists class are free form livings, meaning they live their class in the water or even their own land, and others class are symbiotic, being of course mutualistic or parasitic. And those that are parasitic class, of course, are pathogens of the animal class or the plant. Most reproduce asexually, but of course some are sexual. But of course none have reproductive organs or embryos. As I mentioned earlier, class, most protists are aquatic, hence class. They make up most of the plankton, meaning the phytoplankton and the zooplankton. They are the base of aquatic food webs class, meaning those ecosystems, their class depend on the protists. Without them class, there'll be no bass, no brim, no catfish, no redfish class, meaning no sharks, baby shark or otherwise. I'm kidding. What I'm stating class is the protists are there, and if it's water class, it's there because of the protists, meaning in the ocean, streams, lakes, and ponds. So terrestrial protists are restricted to damp places such as leaf litter, cracks in bark, or even the soil. So shown before you class are Cladiomonas, and it's a photosynthetic organism with two flagella and a cup-shaped chloroplast, as you will all see there, looking kind of green in color. Here class just is an example of protists in a drop of pond water. So with several different modes of locomotion. And this is what you all saw in lab, the amoeba with the pseudopodia, volvox there class, along with Spirogyra. You also saw the paramecium and euglena. I wish we had stentor. Maybe I'll order stentor. And we have the cladiomas. Excuse me, the cladiomonas. So the question is how did protists evolve? Well, it was once stated, class, by this endosymbiotic theory, is that mitochondrial chloroplasts probably originated from endosymbionts, in that certain organelles arose from symbiotic relationships among larger cells, and some bacteria incorporated to live within them. Well, the mitochondria may have originated class as bacteria, and chloroplasts may have originated class as cyanobacteria within a cell. So from that, let's get to prokaryote classification. And I say this to get to, it's evolving, it's changing, ever-changing prokaryote classification is. So if you can take yourself in your textbook to page 544, you'll see just that. Excuse me, 543. Textbook page 543. 543. So there are two types of research that contribute substantially, class, to understanding the phylogenetic relationships among protists. So with those molecular analyses of the gene from the small subunit of the ribosomal RNA and other nuclear genes, and also using the ultrastructure data revealed from electron microscopy. So from such class, we can now get to that we have 5 informal supergroups: the excavates, the chromaloaviolates, the rhizarians, the archaeplastids, and the uniconts. So I won't be here, class, a long time because I'm giving you an overview of the diversity of the protists, but we'll take a bit of time, class, to go through this diversity as it is here. So the excavates class. So we'll be here with euglenoids and diplomonads. So here, class, they have flagella with a deep excavated oral groove, and by way of glycolysis, energy is obtained. Hence, we saw the euglenoids class and the trypanosomes class. This shows a picture, and what's typical, class, is the trypanosomes. Of course, I say trypanosomes because the trypanosomes class are what's here, and typical of which is they are parasitic and, of course, cause disease. You say, what disease? Well, the African sleeping sickness disease by way of a bite from the tsetse fly. So from this, of course, the fever is gotten. So with euglenoids, most are flagellates, and some are photosynthetic, and then some are heterotrophic. So they look just like this. And then, of course, I've already gotten to the trypanosomes. So next up class are the Chromaviolates. So this supergroup class is extremely diverse with few shared characters. And with this, most are photosynthetic. So this includes the dinoflagellates, the apicomplexans, ciliates, water moles, diatoms, brown algae, and even the golden algae. So the aviolates class includes the dinoflagellates, which we had in lab, and apicomplexans. So with this, the dinoflagellates class are the marine plankton. I can't stress enough, class, that these are the basis of those aquatic ecosystems. Without these class, they would not be there. Many of these are bioluminescent class. And what happens sometimes, class, is that billions of these dinoflagellates class produce this orange cloudiness class of the red tide. So these balloons, of course, so these red tides, they're not good. And the reason I said that they're not good, class, is because this can lead to a fish kill because they produce a toxin that attacks the nervous system of fishes. And birds also sometimes die from eating contaminated fish. And it's also been found that even, class, off the coast of Florida, that manatees and dolphins, they have died, and their deaths have been linked right back to these red tides. With the apicomplexans class, well, the apicomplexans, as they are known, are responsible, class, in many cases, class, for what is known as causing malaria. So what happens is the sporozoites, yes, I said the sporozoites, are those small infected agents that are transmitted, class, to the next host by way of the vector. So malaria is caused by an apicomplexan, and as it occurs, the parasite gets into the host, and from said host, class, it's all by way of the mosquito being that vector. So with this, it's, if ever, class, you're going to some place in which malaria is a parasite, or at least malarial disease caused by the parasite, by the vector being the mosquito, you should very well be safe. Up next, class, are the ciliates. So one example, class, would be the paramecium, a type of ciliate that we saw in lab, taking in food here, and eventually, class, using the anophora class to get rid of those that, of course, cannot be digested with the micro and macro nucleus. Water moles, class, once were classified as fungi, but of course you may have heard these being called the fungus-like protists. And of course, they break down decaying matter. Then we have the marine diatoms. So again, these are part of, class, the plankton, found, of course, in the water. And the marine diatoms are quite colorful, and also what's found in diatomaceous earth, which you all may or may not have heard about. Sometimes, class, they can, of course, cause poisoning, class, and even marine deaths. And poisoning, class, would be, of course, in shellfish. Next up, class, is brown algae, and you find this class being of seaweed, which can range up to, class, being a few centimeters to even 75 meters long, such as kelp, brown algae, even the kelp forests in the ocean. Kelp forests. So next up, class, the golden algae, to round out where we are. And with the golden algae class, some biologists group the golden algae with the diatoms, some with brown algae. But of course, there is the golden algae. Next up, class, are the rosarians. So they are monophyletic but quite diverse, including class the forams or the foramina and the echinopods. So these are the chalky ones of which, and they typically, class, settle to the bottom of the ocean and transform into chalk. The echinopods are mostly marine plankton. Next up, class, are the archaeplastids. The archaeplastids includes red algae, green algae, and of course, what's not included here, class, but of course, the land plants. At any rate, class, these are the red algae, and as you're here, going through photosynthesis. So they're a commercially important class for agar, which is used to inoculate bacteria, and even nutrients. Green algae particularly class because, hey, photosynthesis, such as we see here class with what we find on your textbook from page A is Volvox, which is on lab. And then, of course, Ulva at B, and then at C class, Chara, or stonewort, in fresh water. I hope you now see why I say photosynthetic and also include the land plants, even though these aren't land plants. Next up, class are the uniconts. And the uniconts make up what include the amoebas, the plasmodial slime molds, sailor slime molds, and conoflagellates. It also includes the fungi and animals, but of course, we won't necessarily get to those because this is on protists. Alright, so the uniconts class. As they are here, we have the amoebas, which are unicellular, found in soil and water and other organisms. They are asymmetrical and they change their body form continuously, reproducing class asexually by splitting. So that is an amoeba, as you also in lab. The slime mold class may very well have you all think of and remind you of the protists. So if you take yourselves in the textbook from 555 on over to where we find this on page 558, on page 558 at A, we find Plasmodium on that dead log feeding on the bacteria and other microorganisms. And we also find, of course, sporangia, just as, of course, you may have heard of in fungi. And then, of course, those slime molds are there. So next up are the conoflagellates. So they are collared flagellates in the piscithecont clade, which includes fungi and animals. So with this, they are both free-swimming and sessile species, but conoflagellates are of special interest because they're striking resemblance to the collar cells in sponges. In other words, class, this is what provides that supporting evidence between conoflagellates and animals. And what I'm getting at is the comparative DNA sequence data of the mitochondrial and nuclear genes. So it's been put forth that living conoflagellates class and animals probably share a conoflagellate-like ancestor. So there you have it. So what you're seeing here, class, is the very same that is seen, class, in the periphery, the sponges, those, of course, beating collar cells or coenocytes, as previously mentioned. So there you have it, class. This has been the 26th chapter and Principles of Biology 1. So from here, as far as preparing for your final exam with final exam homework, make sure, class, you're able to list and describe the structure and function of 9 eukaryotic cell organelles, including the ribosome. Be sure you're able to describe the central dogma of DNA. Include in each class those processes: replication, transcription, and translation. And I'll ask, of course, about viruses. So you all should know that a virus is not alive. It's an obligate intracellular parasite that cannot be treated with antibiotics. And of course, you should know very well about bacteria and the bacterial shape. Well, not necessarily shape, but bacterial structure and differences in those basic types of bacteria, i.e. archaea bacteria and bacteria themselves, or eubacteria. And then from there, I hope you can understand now why protists are important in those supergroups, meaning that protists represent, class, the basis of all aquatic ecosystems. Without protists, there would be, class, I would say, nothing else in the water. So with that, class, ensure that you're able to contrast both prokaryotic and eukaryotic cells. You should include, class, the types of cells, the structural differences, and functional differences with examples. And we just finished here, class, those supergroups found on page 543, a key point class. A key point. Showing us, of course, that by and large, we find class by way of the uniconts and the archaeplastids, land plants, fungi, animals, and even protists there. So this has been your instructor, Scholar Huff. I'm always here for you, class, so send me an email. Call me on my office telephone number. Stop by after class, before lab, after lab, or even, of course, during office hours. Thank you all.